Welcome to the third lecture of physiology. In this lecture, we will discuss about the nucleus. Nucleus is the most prominent and largest cellular organelle. Most of the cells have only one nucleus. Few types of cells, like skeletal muscle cells, have many nucleus. Generally, the nucleus is located in the center of cell. Structure of nucleus. Nucleus is covered by the membrane called nuclear membrane and contains many components. Major components of nucleus are nucleoplasm, chromatin, and nucleus. Structure of nucleus. Nuclear membrane. Nuclear membrane is double layered and porous in nature. This allows the nucleoplasm to communicate with the cytoplasm. The outer layer of nuclear membrane is continuous with the membrane of endoplasmic reticulum. The space between the two layers of nuclear membrane is continuous with the lumen of endoplasmic reticulum. Pores of the nuclear membrane are guarded by protein molecules. Diameter of the pores is about 80 to 100 nanometer. However, it is decreased to about 7 to 9 nanometer because of the attachment of protein molecules with the periphery of pores. Exchange of materials between nucleoplasm and cytoplasm occurs through these pores. Nucleoplasm. Nucleoplasm is highly viscous fluid that forms the ground substance of the nucleus. It is similar to cytoplasm present outside the nucleus. Nucleoplasm surrounds chromatin and nucleus. It contains dense fibrillar network of proteins called the nuclear matrix and many substances such as nucleotides and enzymes. The nuclear matrix forms the structural framework for organizing chromatin. The soluble liquid part of nucleoplasm is known as nuclear hyaloplasm. Chromatin. Chromatin is thread-like material made up of large molecules of DNA. The DNA molecules are compactly packed with the help of a specialized basic protein called histone. So chromatin is referred as DNA histone complex. It forms the major bulk of nuclear material. DNA is a double helix which wraps around central core of 8 histone molecules to form the fundamental packing unit of chromatin called nucleosome. Nucleosomes are packed together tightly with the help of a histone molecule to form a chromatin fiber. Just before cell division, the chromatin condenses to form chromosome. Chromosomes. Chromosome is a rod-like nuclear structure that carries a complete blueprint of all hereditary characteristics of that species. A chromosome is formed from a single DNA molecule coiled around histone molecules. Each DNA contains many genes. Normally, the chromosomes are not visible in the nucleus under microscope. Only during cell division the chromosomes are visible under microscope. This is because DNA becomes more tightly packed just before cell division, which makes the chromosome visible during the cell division. All the dividing cells of the body except reproductive cells contain 23 pairs of chromosomes. Each pair consists of one chromosome inherited from mother and one from father. The cells with 23 pair of chromosomes are called diploid cells. The reproductive cells called gametes or sex cells contain only 23 single chromosomes. These cells are called haploid cells. Nucleolus. Nucleolus is a small, round, granular structure of the nucleus. Each nucleus contains one or more nucleoli. The nucleolus contains RNA and some proteins, which are similar to those found in ribosome. The RNA is synthesized by 5 different pairs of chromosomes and stored in the nucleolus. Later, it is condensed to form the subunits of ribosomes. All the subunits formed in the nucleolus are transported to cytoplasm through the pores of nuclear membrane. In the cytoplasm, these subunits fuses to form ribosomes, which plays an essential role in the formation of proteins. Function of nucleus. Control of all the cell activities that include metabolism, protein synthesis, growth, and reproduction. Synthesis of RNA. Formation of subunits of ribosomes. Sending genetic instruction to the cytoplasm for protein synthesis through messenger RNA. Control of the cell division through genes. storage of hereditary information, and transformation of this information from one generation of the species to the next. Deoxyribonucleic acid. Deoxyribonucleic acid is a nucleic acid that carries the genetic information. It contains the instruction for the synthesis of protein in the ribosomes. It is present in the nucleus and mitochondria. It is responsible for the formation of RNA. RNA regulates the synthesis of protein by ribosomes. Ribonucleic acid. Ribonucleic acid is a nucleic acid that contains a long chain of nucleotide units. It is similar to DNA but contains ribose instead of deoxyribose. Various functions coded in the gene are carried out in the cytoplasm of the cell by RNA. RNA is formed from DNA. Cell death. There are two processes of cell death: apoptosis, necrosis. Apoptosis. Apoptosis is defined as the natural or programmed death of the cell under genetic control. It is also called the cell suicide since the genes of the cell play a major role in the cell death. This type of programmed cell death is a normal phenomenon and it is essential for normal development of the body. In contrast to necrosis, apoptosis usually does not produce inflammatory reaction in the neighboring tissues. Functional significance of apoptosis. It plays a vital role in the cellular homeostasis. About 10 million cells are produced every day in the human body by mitosis. An equal number of cells die by apoptosis. This helps in cellular homeostasis. It is useful for removal of a cell that is damaged beyond the repair by a virus or a toxin. An essential event during the development and in adult stage. Activation of apoptosis. Withdrawal of positive signals. Positive signals are the signals which are necessary for the long-term survival of the most of the cells. The positive signals are continuously produced by other cells or some chemical stimulants. The example of chemical stimulants are nerve growth factors for neurons, interleukin-2 for cells like lymphocytes. The absence of withdrawal of the positive signals activates apoptosis. Arrival of negative signals. Negative signals are the external or internal stimuli which initiate apoptosis. The negative signals are produced during various events like normal developmental procedures, cellular stress, increase in the concentration of intracellular oxidants, viral infection, damage of DNA, exposure to agents like chemotherapeutic drugs, X-ray, ultraviolet rays, and the death receptor ligands and death receptors. Death receptor ligands are the substances which bind with specific cell membrane receptors and initiate the process of apoptosis. The common death receptor ligands are tumor necrosis factors and Fas ligands. Death receptors are the cell membrane receptors which receive the death receptor ligands. Well-characterized death receptors are TNF receptor 1 and TNF-related apoptosis-inducing ligands, receptors called DR4 and DR5. Role of mitochondria in apoptosis. External or internal stimuli initiate apoptosis by activating the proteases called caspases. Normally, caspases are suppressed by the inhibitor protein called apoptosis-inhibiting factor. When the cell receives the apoptotic stimulus, mitochondria releases two protein materials. First one is cytochrome C and the second protein is called second mitochondria-derived activator of capsaicin. Apoptotic process. First, activated capsaicin digests the proteins of cytoskeleton and the cell shrinks and becomes round. Because of shrinkage, the cell loses the contact with neighboring cells or surrounding matrix. Chromatin in the nucleus undergoes degradation and condensation. Nuclear membrane becomes discontinuous and the DNA inside nucleus is cleaved into small fragments. Following the degradation of DNA, the nucleus breaks into many discrete nucleosomal units, which are also called chromatin bodies. Cell membrane breaks and shows bubbled appearance. Finally, the cell breaks into several fragments containing intracellular materials, including chromatin bodies and organelles of the cell. Such cellular fragments are called vesicles, or apoptotic bodies. Apoptotic bodies are engulfed by phagocytosis and dendritic cells. Apoptotic bodies are engulfed by phagocytes and dendritic cells. Abnormal apoptosis. Common abnormalities due to much apoptosis: ischemic related injuries, autoimmune diseases like hemolytic anemia, thrombocytopenia, AIDS, neurodegenerative diseases like Alzheimer's disease. Common abnormalities due to little apoptosis: it is cancer, autoimmune lymphoproliferative syndrome. Necrosis. Necrosis means death in Greek. It is uncontrolled and unprogrammed death of the cell due to unexpected and accidental damage. It is also called cell murder because the cell is killed by extracellular or external events. After necrosis, the harmful chemical substances released from the dead cell cause damage and inflammation of neighboring tissues. Causes of necrosis. Common causes of necrosis are injury, infection, inflammation, infarction, and cancer. Necrosis is induced by both physical and chemical events such as heat, radiation, trauma, hypoxia due to lack of blood flow, and exposure to toxins. Necrotic process. Cell swells causing damage of the cell membrane and appearance of many holes in the membrane. Intracellular contents leak out into the surrounding environment. Intracellular environment is altered. Simultaneously, large amount of calcium ions are released by the damaged mitochondria and other organelles. Presence of calcium ions drastically affect the organization and activities of proteins in the intracellular components. Calcium ions also induce release of toxic material that activate the lysosomal enzyme. Lysosomal enzymes cause degradation of cellular components, and the cell is totally disassembled, resulting in death. Products broken down from the disassembled cell are ingested by neighboring cells. Reaction of neighboring tissues after necrosis. Dilatation of capillaries in the region and thereby increasing local blood flow. Increase in the temperature leading to reddening of the tissue. Release of histamine from these tissues which induces pain in the affected area. Migration of leukocytes and macrophages from blood to the affected area because of increased capillary permeability. Movement of water from blood into the tissue causing local edema. Engulfing and digestion of cellular debris and foreign materials like bacteria by leukocytes and macrophages. Activation of immune system resulting in the removal of foreign materials. Formation of pus by dead leukocytes during this process. Finally, tissue growth in the area and wound healing. Thank you.